Hi class! In this online module, we are going to be learning about Punnett squares. Punnett squares can sometimes be tough in the beginning, but once you get the grasp of it, it will be as easy as 1, 2, 3. The value of Mendelian genetics is being able to predict the results of a particular trait in a mating of two organisms. The probability of a trait can be determined using a Punnett square to expose all possible allele combinations. Before we go on to making Punnett squares, there are a couple of terms we need to keep in mind. An allele, as we defined before, is a different form of the same gene that resides at the same locus on homologous chromosomes. Dominant alleles are when an organism has two different alleles for a trait and only one allele is expressed, masking the effect of the other allele. And recessive allele is the masked allele. In order for an organism to express a trait, there must be two copies of the allele present. When making Punnett squares, first you want to signify the dominant allele using a capital letter. This is usually the first letter of the dominant allele. So in this example, we'll use P for purple flowers. The recessive allele is then symbolized by a lowercase version of the same letter, thus a lowercase p for white flowers. Next, we want to determine the genotype of the parents. Remember, the genotype is the genetic complement and shows the alleles present for an individual, such as two capitalized P's for purple flowers. The genotype can be homozygous, meaning that both alleles are identical, such as two capitalized P's, or two lowercase p's, or it could be heterozygous, meaning that the alleles are different, such as a capital P and a lowercase p. Heterozygous organisms are considered to be carriers of the recessive trait, but do not express the recessive trait. Carriers may not even know they are carrying a particular trait. So, if the genotype is homozygous dominant for one parent and heterozygous for the other, both produce purple flowers. The homozygous recessive genotype is the only genotype that produces white flowers. The phenotype for an organism is what trait is expressed by the alleles, either purple or white flowers in this example. To construct a Punnett square, you first want to separate the alleles and place the genotype of the female in the top section of the square and the male genotype along the left side. In this example, we are using P to represent purple flowers. So the female is heterozygous and the male is homozygous dominant. For the female, place one of her alleles in the two squares directly beneath her allele possibilities. For the male, spread each allele to the right and fill in the two boxes. Once complete, there should be two genes in each box, one from each parent. Follow this step-by-step diagram if you need assistance. Looking at our final box in the diagram, we can record the possible genotypes and the number of potential offspring with each genotype and convert it to a percent. So in this case, there is a 25% chance of homozygous dominant genotype, a 50% chance of heterozygous genotype, and a 25% chance of homozygous recessive genotype. So there is a 1 to 2 to 1 ratio. However, the phenotypic ratio is 3 to 1, as 3 of those genotypes have at least one dominant allele for the color purple. Thus, they will all express the purple color, and only one has two recessive alleles for the color white and will express white coloring. We will be going over this more in class tomorrow, but start to practice it now. There will be a website and practice problems to help at the end of this PowerPoint. There are some inheritance patterns that do not follow the dominant recessive rule. Non-Mendelian inheritance relates to the patterns Mendel did not know. Incomplete dominance is one of those patterns. Incomplete dominance occurs when neither allele is strong enough to dominate the other allele. Thus, there is no dominant or recessive allele. Both genes are expressed in a blended fashion. A common example of this is seen when a certain red flower crosses with a white flower to produce a pink flower. In the diagram, you can see neither the red nor the white allele is dominant or recessive, so a blending of the alleles occurs. Now this diagram takes the pattern one step further and crosses two pink flowers. And you see the offspring depicted in the Punnett square. Usually for incomplete dominance, a capitalized C is used to signify incomplete dominance, and the letter you want to label for the trait is seen as a superscript above the capitalized C. So A white flower is capitalized C, superscript capitalized W, a capitalized C, and another superscript capitalized W. A red flower is capitalized C, superscript capital R, capital C, superscript capital R. And the pink would be capital C, superscript capital W, capital C, superscript capital R. In the cross between two pink flowers, you can see the F2 generation offspring has a 25% chance of being red, a 50% chance of being pink, and a 25% chance of being white. We will be doing more practice with incomplete dominance in class. Another non-Mendelian inheritance pattern is codominance. Codominance is similar to incomplete dominance except for the fact that in codominance, both alleles are displayed at the same time, creating a third type of genotype with no blending. There is no dominant or recessive allele because both alleles are expressed together. In the phenotype of the offspring, a common example of this is human blood types. A person with type AB blood has alleles for both type A and type B. Neither is dominant nor recessive, but both are displayed. Another example is a speckled chicken that has both black and white feathers. In the example shown of the speckled chicken, you can see that it is given the genotype capital B, capital W to encompass both the black and white colors. Polygenic inheritance is also considered to be a non-Mendelian inheritance pattern. It occurs when multiple sets of genes produce a trait so that the more genes that are inherited for a trait, the greater the expression of the trait. Many human traits such as height, weight, hair color, eye color, skin color are influenced by polygenes. For example, the more genes a person possesses for dark hair, the darker their hair. Sex-linked genes are another form of non-Mendelian inheritance. There are certain genes located on the sex chromosomes that may be involved in sex-linked inheritance patterns. Remember, the genotypes for females are XX and for males, XY. For a recessive gene to be expressed in a female, it has to be present on both chromosomes. However, for a male, it may only be required to be expressed on the X chromosome. For example, color blindness is a sex-linked recessive gene that is present on the X chromosome. The Y chromosome contains no gene for color blindness. Females have a lower frequency of color blindness because they have to receive the recessive gene from both parents. However, if males inherit the recessive gene for color blindness from their mother, they will be colorblind. There are several more patterns of non-Mendelian inheritance that we will be covering further in class. Punnett squares take practice. Use the website provided to help. It's a little silly, but it will definitely help you if you are confused. Here are some Punnett square practice questions I would like you to complete on a separate sheet of paper showing all of your work and bring it to class tomorrow to go over.